Sir,
Yes.
you live now, sir.
All right. All right. Very good evening, everyone. All the students who are joining us today for the second live session of MOOC course on Bioengineering and Interactomics. Welcome to today's live session. Of course, in live session, we are available to talk to you and take your queries. But also we thought to make it more thematic and give you idea for some of the cutting edge state-of-the-art technologies which are used in proteomics and of course in the integrated omics technologies. So we're going to focus on microarrays and label-free technology. Along with me, there will be the course TL as well. And we'll try to give you the flavor of these technology, not too much on the basic side, but rather more from the lab about the demonstration and some of the experiments. Let me just give you a quick overview of the protein microarrays. Of course, some of you have already studied in the Interactomics course. Those who have not studied this technology, these are miniaturized glass slides on which, if you think about a simple situation that you have printed the antibodies, these are purified antibodies which have been printed on the glass slide, and let's say you have 100 of those antibodies printed. Now you want to know for those proteins for which you have antibody, what is the abundance of that particular protein from the given sample, and you have to screen that. So antibody is pretty much used for the abundance-based proteomics. Whereas if you have to look for more functional information, so targeted protein array approaches are being used. Especially you can have the protein as a purified form or take from the cell lysates, tissue lysates, phos products, or sometimes people also take the cDNA directly and add the in vitro transcription and translation method to look for the making the proteins on the chip. So broadly there could be two types of arrays, antibody-based arrays more on the abundance screening, and functional protein arrays, peptide arrays more for the functional analysis. This is a broad classification. These can be used for many applications, protein microarrays and peptide microarrays, especially biomarker identification is one of the key areas when people try to look for, but given that these are small slides, we are going to have the very small volume of the patient sample. With very few microliter of the serum or plasma sample in dilution, you can actually spread them on the entire chip. So you can do that. Additionally, if you have already seen the mass spectrometry technology in the past, when you want to process a sample from the mass spec dynamic range of the instrument is quite limited, 10 to the power 5 or so. Whereas when you think about the serum or plasma sample of the blood, they are having the very huge abundance difference from the low abundant to the high abundant proteins present in those samples, which could be to the tune of almost 10 to the power 11. So now all the proteins cannot be written in the mass spec, right? Given the limited dynamic range. So you have to deplete out the high abundant proteins from the plasma and serum sample, and then only you can run in the mass spectrometer. So that's one of the challenge in the doing this kind of biofluid with the mass spectrometer. However, here in the protein arrays, we are looking at the directly we are without any depletion, we are probing the samples on the chip, and we are going to look for the fluorescence signal, which can be then used to quantify the signals. So this can be another powerful application here, given that you are not changing anything the nature of the sample by depletion. You are not removing many of the abundant protein, which might be carrier of the important biomarkers as well. So therefore, you have much better way of identifying the biomarkers with very small volume using the protein arrays. This is what shows in the top panel of the slide. Additionally, if you want to look at whether a protein X interacts with protein Y, protein interacts with the small molecules, nucleic acids, carbohydrates, you can again look for these kind of interaction studies. Or if the protein gets phosphorylated or glycosylated, could also be tested out using these kind of array platforms. So there could be n number of applications using this technology. How we use this technology in our lab, let me just walk you through with two case studies. One about the cancer. So in cancer, like in general in our body, if there is a cancer cell is arising, body's immune system senses that as a foreign cell because there is something suspicious going on, where uncontrolled growth is happening. And therefore it starts producing some autoantibodies to control that response. These antibodies are very stable, So therefore, can we identify those autoantibodies produced in our own body against the cancer cell ahead of time and can be utilized for the screening? So that kind of hypothesis for which we and other labs, they use the UProt arrays like human proteome arrays. Idea is can we have all the human proteins printed on these kind of glass slides and then use those the chip to screen for the potential autoantibody biomarkers from the biofluid of these patient samples. So we did this study with the Chatar Memorial Hospital to look at all the brain tumor gliomas patients. The workflow is very simple. You are having these kind of protein arrays. You are blocking them with the milk or the super block so that all the, other than the protein spot, rest of the array surface is blocked with this molecule so that no non-specific binding can be seen. Then you incubate that, after cleaning it up, then you incubate that with the patient serum sample. If the patient is having the antibodies for the cancer, it can actually bind over there on the chip with the antigens. And then you are going to use the anti-human IgG or IgM and probe them with a secondary conjugate fluorophores and then look for the signal detection with the microarray scanner. So this kind of the broad workflow for any kind of protein microarray study. What we use here in the this particular study on the glioma, we took grade 2, grade 3 and grade 4 patients and try to screen for whether any pattern exists if you move from the low grade to the high grade of the glioma patients. So after looking at the entire data and doing series of data processing step and normalization step, we found in grade 2 there were very few number of autoantibodies. In grade 3 quite large number of autoantibodies and grade 4 again the numbers got reduced. If you think about the actual disease biology here, given in the brain tumor there is a blood-brain barrier, so very few autoantibodies were expected to be seen in the low grade. And as it became the aggressive form, grade 3, the blood-brain barrier reaches out and most of the antibodies are seen that time. And grade 4 glioblastoma when most of the things are dying right at that stage. So again the number got reduced. So we could see that some proteins are showing the pattern, like using the microarrays, one could see how a response can be seen from the low grade to the high grade. And in this case the signals are going down, IgG1 again signals are going up. So can we find the pattern of these autoantibodies which can be detected at a very early stage from the control to grade 2 or grade 3 and then we see the progressive changes happening in the signal intensity. So based on this we propose this pair of marker which is recently patented as well and we are further testing and working on this. Additionally we also looked at colorectal cancer and we could here see some different pattern of the autoantibody marker, not the same what we saw in the brain tumor. Here the healthy is very showing no response and different cohorts or different patient groups are showing a good signal for these autoantibodies. So again, these kind of methods can be utilized for different type of cancer screening and different type of other autoimmune disorder screening. Let me switch the gears here. So in addition to the protein microarray which I talked just now, which was the full-length protein purified, and now we are trying to screen for the biomarker. In this case now we are going to use the peptide microarrays. And think about the COVID-19 disease scenario and the virus SARS-CoV-2. So for this particular virus, for all its protein, all the peptides were synthesized synthetically, and they were printed on the chip, and now this chip became peptide microarray. So the idea was, can we take the severe and non-severe group of the COVID-19 patients? We have this serum sample from these individuals and the plasma samples, which we did a pre-heat treatment to ensure that the viral things are inactivated. And now when we are going to probe them with the secondary antibody with the anti-human IgG and look for the again signal intensities. So this entire workflow, the D panel, requires a lot of steps which one could follow for doing the data processing in the microarray. And for that, shortly I will have a session with another TA who is going to walk you through the series of steps involved from the scanning till the data analysis part. So what we found from this study, we could look at eventually that there are some specific patterns emerging when we look at IgG or IgA response, that some of the immunogenic peptides across all these patients which one could try to locate and then see where these peptides are actually found in the full-length proteins of the spike protein or nuclear protein. And this information could be helpful eventually to look for the new vaccine design and identifying the potential immunogenic regions for these peptides. So how these experiments are done? So let's now, I have already talked to you a lot of theoretical part and given you some idea for the applications. Let me now show you this video of the steps involved in doing this kind of work of the peptide microarray, and then we'll take you to the lab to show you the actual setup followed by the data analysis part. So let me just show you this video.
Today, we will be demonstrating peptide microarray technology. The first step involves reagent preparation, namely washing buffer, staining buffer, and Rockland brown buffer. After preparing the reagent, we move on to incubation tray assembly. Unscrew the incubation tray by slowly turning the screws diagonally. We will be using two black brass clients as balancers, since today we will be using only one microarray chip. This is the pepperprint microarray chip. Slowly take out the chip using plastic forceps and place it on the incubation tray. Align the chips properly onto the tray. Now, allow the gasket to be placed over the slides. Remember to keep the glossy side of the gasket on the downward side. Align the silicone gasket over the slide and tightly seal the incubation chamber using the screw. Remember, tighten the screws only diagonally. This will ensure that the slides do not break. Next, add 2 ml of staining buffer onto the chips. This step is called equilibration of peptide microarray chip. Please note that we are not touching the microarray chip at any point. Now allow the equilibration process to happen for 15 minutes. Aspirate and remove the buffer and add 2 ml of blocking buffer. Slowly add the buffer onto the slides. Allow for 30 minutes of incubation. The next step is sample incubation. To the staining buffer, we will add patient plasma at a dilution of 1:200. This is the patient plasma. Now slowly overlay the diluted plasma over the slide and incubate overnight. This will be done in a cold room at 4 degrees Celsius at 140 degrees. At 140 RPM on an orbital shaker. On the next day, remove the incubation tray from 4 degrees Celsius and aspirate the solution. Now add washing buffer thrice onto the chip. This step is done 3 times in the dark. Again, completely aspirate washing buffer and add secondary antibody. Prepare for dilution of secondary antibody 1:2000 and then slowly overlay the secondary antibody on the microarray chip. For 45 minutes, allow the incubation to happen at 140 rpm at room temperature. Make sure that you do not expose the slides to light at any given moment. Now remove the buffer and wash the slide thrice using washing buffer. Now gently take out the slide and put it in dipping buffer. This is done to wash away all the salts that may be over the peptide chip. Use a pressurized stream of nitrogen to completely dry the slides. Finally, we will scan the slide. This is the microarray scanner, GenePix 4000B. We are using manufacturer recommended settings for scanning the slide.
Alright, so I'll stop it here because from here we'll take it live. I request our colleague to share his screen, and he's going to now show you the setup of protein microarray, peptide microarray scanning, as well as data, at least some step of data analysis processing, which will be shown over there. And followed by Amrita will then talk about SMC Pro for the immunoassay development. So these two will be next live session will have. All right, Surabhi and Alka, please take over.
Right. So as you saw the procedure of the microarray experiment, we are going to demonstrate the setup as well as the slides live. So these are the slides which I have. These are called the microarray slides. This one is a protein microarray slide. It has all the proteins printed, all the human proteins printed on it. And we also have another microarray slide, which is the peptide microarray slide, the one that was shown recently. This is the peptide microarray slide, and it has the peptides of SARS-CoV-2 virus printed on them. So, as per the protocol shown in the video, these slides are incubated with serum samples. So, antibodies from the serum sample, like the IgG, IgA, and IgA, can bind to the proteins or peptides on the slide. And the antibodies in the serum are further closed with secondary antibodies, which are fluorescently packed. So when these fluorescently antibodies, the fluorescence from those probes are measured in the microarray setup. So this is the microarray setup. My colleague will demonstrate the microarray scanner and the extraction run.
Hello everyone, my name is Arthur. So this is the first slide. I already started experiment to be just explained about it. Now I'm going to scan a particular slide in the scan. So this is the scan. This is on specific slide. So now this is opening part, and you can see that this is opening. So this slide already inserted into the scanner. Now we need to scan this particular slide to see the specific signal. So there are importantly, sophisticated softwares are there in the system. We are usually use Genfix Pro for this scanning purpose. After the data extraction part, we usually follow this, use this Genfix Pro 7 software. Now I want to show you how we usually use this one. So this is Genfix Pro 7. So now you can see this is searching for the scanner. So first of all, as already sir mentioned regarding this Genfix specifically required. So for the QC, we usually go for this 530 nanometer for this quality checking of the chip. Followed by this functional antibody screening, we usually go for the 635 nanometer setting. That is the red laser, 532 for the green laser. So first we need to know these settings. So here you can see this hardware settings is to be done. So there we need to first initialize this PMTN. This is a very important parameter followed by the laser power that we need to set first. Then we need to show this previous scan. Performance is about the previous scan because if there is any problem with this, let's say for this specific experiment, after this previous scan, at least we can go to the second set of the experiment for the specific site. So this check is it will take some time. So better I will show you how to extract the data for the specific microarrays as well as the data window. Microarrays is for protein arrays and the protein arrays for the peptide array. So now I'm going to the another software that I already mentioned, that is Genfix Pro 7. Now I will give you very minute details about this software and how you can set these settings. So first of all, you can see this images. So this is the macroarray image. Now first, we usually go for the scanning specifically in the QC part. So for the QC, we need to look into some GST tracked proteins. So how we are getting this linear trend or not, as we usually get it after linear trend. So this would be the specific scores that we need to check this after getting the data. So now how we can go for the data extraction. So one of the important file that is required, that is called the GAN file, that is for Genfix arrays. How this looks like. Now I just need to place into that because it's already extracted, it is little bit time consuming, so you can see some kind of circular grids into the specific spots. So this will actually help us to extract the data. Now this is just specifically the GST tag protein, so now you can see after getting the data, are you getting a linear trend on it. For the QC check, we usually prefer for the protein arrays this the settings and followed by the green channel. But this is actually the grids you can see, this is called the Genfix analysis part. Now, this is actually time consuming. Sometimes this not be very uniform, so we need to specifically extract this particular expand specific feature indicator because this is technically from this popular feature indicator. That is sometimes need to expand, sometimes contract because of this intensity of this cost. After the QC passed, then we move forward to the rate channel. Now showing about everything for overheads. Now this is the same chip where is scanned for this rate channel of this settings of the laser. So there you can see this is this cost. So it might be this is the very intense signal you can find, might be this marker, but obviously this will we can only understand after we can analysis. This is not test analysis. So how you can place this box? So you can see this image. This is the image. Also you can just place on this view box. Then you can place this base properly onto it. Now this block will be also adjusted properly like this. You can see this is kind of a sequence. You can do this shifting of this block as well, specifically whatever required. And feature indicators is also could be while changing the settings. So now you can see this could be also expanded properly after we use this settings. Now, after this, we usually go for the extraction of the data. So for that, we need to go for this section of the results. So then we can find this data scan. So if we just place onto this data scan, so you can see this is the blocks. So there are a lot of blocks that are shown in this give you some demonstration of this. Now you can see this is the creating event as well as the process channel. Now you can see. So this is this block one. This is the column row and what are the associated proteins and proteins is showing. Medium of the mean value of the foreground, background, everything is coming here. And this is actually we take it as the GPF file. So how the GPF file also looks like? Just like this. This is a CSV file. So this is what GPF file I am showing. So this GPF file is also giving us finally the GPF file usually take the data analysis into understanding the data. Now I have already shown you just this open file, but something also should be mentioned about the peptide as well. So how this peptide also look like? These images looks like. You can say this is what peptide is. So if this peptide you can get the deviation because last time we got the circular features. This time this is kind of a rectangular shape, better to say oval shape. So this is kind of a peptide. Some also might mention about the immunogenic peptides, immunogenic epitope. So there you can see this is kind of a intense signal message. So contiguous peptides, you are getting some signal contiguous. So this is one peptide is a duplicate. So contiguous are getting a very good signal. After analysis, if we are claiming so this is might be one peptide epitope. So this could be a epitope. That means it is a contiguous peptides we are getting claiming as immunogen. So we also we have experienced we developed the graph form. So this graph is little bit different. So you can see how kind of this kind of features like this more or less this rectangular shape. So this is also you can extract the data after placing the bits onto the specific spots. And finally in the same, so this is for the IgG. Now the IgG looks like the association. So this is for the red channel. So for green channel, for red channel also you can find also this specific just yes. So this is the image. So this would be also epitope. Maybe if I go for the analysis, also we can after we can say this could be an epitope. So after this epitope, this specific images and placement of the bits. So this is the bit that we have developed in the lab. So now after that again we go to the other section and go to the this we just place this data. So it will generate the data automatically. You can see this also but yes, I mentioned regarding the loss, so that I have to tell you what the loss. So now you can see this particular image. Different this one, this is block 1, this is block 2. This one, when the data is also generated, which is showing this block 1, block 2 in this manner. So in this peptide, we have different number of blocks, but in the protein and peptide also. In this way, we have prepared this and go for the data extraction. So after this data extraction, actually there are a lot of steps associated with this data analysis part. And majorly today we just wanted to show you some of the demonstration of this data extraction. So maybe later stage I will give you a broad idea about the data analysis, how we can integrate that into the other language, the statistical analysis, and finally getting this analysis. So that's all for our side. Thank you. And this.
All right, thank you, Arka and Surbi. Amrita, maybe you can now talk briefly about the immunoassay and show the some process.
Hi everyone. So today we are going to show you or demonstrate briefly about a very high throughput immunoassay technique, which will be done by this particular instrument called SNC Pro. SNC Pro is single molecular counting flow instrument. So what it does is that it's an ultra sensitive immunoassay technique that can detect any molecular marker or any protein in a very small amount which is present in a clinical sample. So for example, you are trying to find a particular protein which usually is getting masked by any other abundant protein in your clinical sample. But here, using this particular instrument, you are going to, or this particular assay, you are going to, you are able to detect the levels of this particular protein in a very low amount as low as femtograms amount. So now this instrument works on the beat capture immunoassay. So for example, onto a bead, and then the beads are placed onto a surface of well, and you pour your clinical sample, complex clinical sample onto the well, and the antigen, the interaction happens, the immune interactions happen, and then once you go on for a wash step where any unnecessary interactions are removed, and then finally you elute out those immune complexes, which is the antibody and your sample of interest and your protein of interest, and you then place that elute onto this particular plate. So this particular plate is a 384 well plate. You place your samples over here, and here I'm going to show you the application where you monitor the experiment as well as you run the analysis. So here we are going to open the instrument and place the particular plate here. So as you can see, I'm placing the plate here in the instrument, and then we close the lid, and then we run the analysis. Now, as you can see, this is the software expert that does the analysis. Here it's the layout of every well. It's a 365 well layout. You can first use your standard samples to make a standard curve, and then you can place your own samples having multiple samples at a time, which allows you to do multiplexing. That means in one go you can analyze and detect very small amount of proteins of more than hundreds of samples. Now once you run the analysis, it will take couple of minutes to run the analysis. Here we are going to show you a demo data where we have run our standards to generate a standard curve. As you can see, this is the standard curve we can generate, and these are the concentrations that we have gained from the standard curve, that is from the picogram per ml amount. And so basically this is how this method is advanced from the other immunoassay methods. Number one, sensitivity. So it can detect the protein changes, molecular changes at very low concentrations. Then it is faster. It can detect it's a very robust and fast techniques in other immunoassays where you have to do the experiments for like days, couple of days, and ELISA and other techniques. It will only take the sample preparation time, and the analysis time is very low. And number three is multiplexing, where you can run multiple samples at one go. So that's it from my end. So my colleague Nature will give you demonstration of another macromolecular interaction technique.
All right, thanks, Amrita. Let's now move on briefly to the label-free technology. So what we have been talking so far were essentially microarray-based platforms, and signal detection were done using some sort of fluorescent-based tagging. Now, like most of the applications which we talk, you can get an answer about the binding. You can get an answer about, some interactions are happening or not happening, and what kind of biomarker signal you can see from the chip, right? But in all of them, as I said, you have to use some way of a signal detection system, maybe fluorescent-based is the most commonly being used. Other type of HRP-based and other immunoassay-based, signaling methods can also be utilized. Radio labeling and other stuff, which is not so commonly used. But any type of labeling is actually going to introduce some artifacts. The challenge over there will be that you are going to your labeling strategy is going to remove some of the coding aspect of the proteins, and with those chemical interferences, you always have some artifact for the binding. So while you can see yes or no answer from the microarray fluorescent-based technology, but you cannot get very clear binding or kinetic data from that activity. So therefore, many type of label-free biosensors are being utilized. One of them is surface plasmon resonance, which is very commonly used technology. Here we have, a prism, a gold plate, a light source, and then we have, let's say, you are having antibodies immobilized on the gold chip, and now you are flowing the protein, and as soon as that protein and the antibody pair binds, you will see that, okay, now there is some sort of binding is being seen, that is known as association rate. After some time, you will reach to saturation state, and you keep flowing, and then molecules start coming out of it. That's where you can see now the dissociation state or the K small d. This is just one other example. You can always have the protein-protein interaction, protein small molecule interaction. The binding mechanism will remain same, and what you are watching is here SPR sensorgram with the time on the x-axis with the response on the y-axis. And here, since you are generating that a binding is happening, but also with what type of on rate, what type of off rate, you can actually generate the kinetic data looking at the K capital D or the diffusion constant for that particular type of binding. So this is quite important way of looking at the biomolecular interactions, because you're not labeling your protein, but rather you are directly trying to look at the percentage change in the reflectivity of the medium, and as a result you are looking at whether two molecules are binding or not. So it is indirect method, but it has more advantages sometimes over the fluorescence method, because the signals could be much more real here, and you are not changing any kind of alteration, you are not doing anything from outside for the binding to happen. There are many types of platforms. I just talked to you about SPR, surface proton resonance. Other is nano-hole arrays. Carbon nanotubes are also being used. Planar wave guide arrays, nano-wire sensor arrays, SRIB, spectral reflectance imaging biosensor. These are different principles involved. Idea is we are looking at just change of the two molecular binding with the dielectric constant change, with the reflectivity change, with the reflectance change. And likewise, we are going to now talk to you shortly about the biolayer interferometry based method, which is going to be demonstrated directly from the lab by Nirjit. He's going to show you this instrument which works on the another principle, not with like the SPR with the reflectivity, but here we are looking at the how you can measure the things in solution format. You are not going to immobilize on the gold chip format which I just showed to you. So over to you, Nirjit, to give you some demonstration about the BLI technology.
I hope I so bilayer interactometry. So here basically what we do is all sorts of interactions, that protein antibody. We have so all those kinds of interactions, whatever you want to study with the instrument. So as you can see, there are two compartments for that. One is where the samples, your ligands and the analytes, and the other is where the sensor, the main workhorse of this instrument is. So as you can see, the sensor is very delicate in that sense, and I'll first say about some things of how the instrument works. As you can see here, white light incidence from this region to each of the sensors that gets attached to it. So now that instrument analyzes the interference of the white light that gets reflected back to two surfaces. One that is the only surface of this sensor, this space only, and the second one with each molecule that gets bind one layer after the other. So first, the sensor, based on the sensor chemistry that we can use streptavidin sensors, and then we can take ligands which is our biotin ligand, and similarly many other chemistries are there we can take accordingly. So first, one ligand, one of the two interactors, which requires the ligand is found with the sensor, and then that ligand goes into interacting with supposedly one of the molecules which we call the analyte. So that's how we can monitor multiple concentrations of the same from one analyte with a particular concentration of a ligand. So this is a robotic arm of this instrument. So it can move around in one direction, in this direction. So each samples passes can be pushed together. And let us move towards the software. So that's how we have to define all the things starting from the plate map. So L refers to the ligand, the one which we are putting our first interactor and all those samples which are basically analyzed, where we put out all the second interactors, depending on the different chemistry. So we have to define the concentrations, and also we have to mention the different states, starting from baseline to loading. Loading is basically loading of the ligand, the first interactor, and also the different dissociation and association steps. So this dissociation and association step, that's what gives us the Ka and Kd, the K off or K on, whatever you want to call it, and that's how the calculation of the actual Kd gets into mapping. So again, coming back to, so we have to also show the sensor alignments, and that's how the samples are run. I'll now show you one of the run samples, how it looks like. So this is a standard samples run in the kinetics mode. So we can also do both kinetics as well as some quantification. So absolute or relative quantification. Also there are epitope pinning and other strategies as well. So this is just a kinetics experiment just to check the KD values of two interacting partners. So as you can see, we can load up the sample here. Here we have taken biphenylated Pro A sample on a stripped evidence sensor and interacting it with ice. So here, as you can see, this is the sensor. So we can also mention which is the reference sensor, basically the negative sensor, where nothing has been loaded, no ligand has been loaded. So that also gets to the effect, and we can subtract that. Similarly, we also keep a zero concentration sample analyte, which also gets subtracted from the other concentration, and some of the data correction steps. Finally, moving on to the kinetic analysis, if we want only the association or both association dissociation or only the dissociation into the account, how do our interactors interact with each other? Is it a 1:1 model or some other models, and one molecule acts with one molecule only, or so on and so forth. So all the other parameters are also there. Finally, we can get the desired information about the Kd and the Ka values, and thereby the Kd, the final concentrate, the function values, the rate values that we can take into account of the kinetics of those two interacting partners. So this was majorly what things can be done. And now, as you can understand, based on the different applications, there are several other sensors, biosensors. Some are storage sensors, some are GSP tax sensors, some are any other amide sensors. There's also experiments, those can be done. We wanted to study interactions related to multiple questions. So that in a level field.
All right, great. So thank you, Richard, for showing the instrument and briefly talking about in which way one could utilize this particular setup. Again, like since it is done in solution form, it has its own advantages over the other type of biosensors, because now you are actually measuring the things in the solution format. Also, the same solution or the biofluid or the sample drug molecule can be reused for the other type of interaction studies in the later on part. So now, since we have talked about few things regarding the biosensors and the different type of data sets, I just want to give you the broad picture that eventually from all of this technology, what we want to achieve, we want to get much more comprehensive picture which can be helpful for understanding the biology. So in our case, we are not generating genome data or transcriptome data, but it is, I think, good if you have available data set or if you can generate data. It's a good idea to have the multiple layer of information generated. But essentially, if you are looking at even the more dynamic molecule like proteins and metabolites, environmental impact, how to take all those cues and the clues available and try to make use of them, integrate them to find out what is the best interacting partners, what is the best pathway which is emerging as a network, which is critical for that type of problem or the given disease or looking at the drug treatment. So that's where a lot of bioinformatics based workflows are important. So next we are going to show you some of the key tools which are available, which are mostly available online, which can be utilized to analyze your data and give you some better visualization for looking at the interaction networks or looking at the pathways. So now Ankit is going to take this session for you.
So thank you, sir. So I'll just share my screen. So we'll be looking into the network analysis. So now you have gone through as well as given a brief overview about the different interactions, also starting from different interactions to ways we have understood, right from starting from the autoantibodies to the immunogenic peptides. But the major concept behind omics or this kind of high throughput technologies is to understand at the whole systems level, as Sir was mentioning, and the understanding at the systems level means we all the proteins or the metabolites at a large scale, it remains connected and also influences each other's information. Is that what we can understand? So one of the major goal of this kind of studying interactomics or interactions or omics as a whole is to understand their interaction and what are the changes that occur mostly in this kind of in a body, and when there is a change in the disease level, so what is actually happening. So this is an excellent article that you all can go through, which tells us about how there are only among the all the genes on the some of the genes which are associated with diseases, and some of the are belongs to the essential among those which are some of them are essential as well. But there are many genes which are essential and which are not related to the diseases. So how they are connected with each other and how they are which are leading to the disease associated protein further from the genes level can be studied through a interaction map. So now if we try to find out in the mode or module for understanding the network, there are different modules can be done, and it is very much can be different graph theories in the mathematics to understand that how these genes are interacting with each other or the proteins are interacting with each other, and what is their directionality and what is their influence over each other to understand our disease system very well. So, for example, the topological module which says about that the closely related genes. So, if one closely related gene is affected, then what is the effect on the next closely related effect, or the one in the analog level or the functional level. That if the two genes are working towards the same function, for example, if two genes are working on a phosphorylation or post-translation modification of a protein and gets affected, then what is the further role in it in a disease thing. Another is the disease module, where there are different proteins which are associated only in certain specific diseases and can take us to a directed graph kind of network, which we can give a directionality to it to better understand. So these are some of the modules or some of those interactive networks of what we visualize, like the interactive reconstruction or the disease module identification among the disease and the protein. This will lead us to understand at the cellular level in a much better way for targeting for newer drugs you can target for blocking those major hub or suppressing or activating those enzymes or those essential important proteins which cascade a lot of the functional other signaling pathways, or to understand the pathways, and we can have in understanding through different homogeneity and dynamic homogeneity and the different Ds, as well as the linkage methods or the disease module methods, and the other kind of methods can be used. So this is how we can go through to understand the human disease network. So like different genes, for example, this was an example of a breast cancer with the different genes, and how it is also in the bone and cartilage cancer. So see, you can see that there are certain common genes and which makes an effect, as well as the metabolic network. So this is what a typical biological network is looks like in a metabolic scale. So you have the proteins, the genes, the metabolites, the PPIs, that is the protein-protein interactions, the genetic interactions, or the gene regulations, and the cell signaling and the cell metabolism. So all these, if we have the data through the high throughput techniques from both microarray and the mass spec based techniques, we can construct this kind of network to understand the system
level well. So one, we have the physical interaction network, another is the functional association network. So one is that is a co-expression genetic network, and the physical network interactions are the CPI signaling network and the those networks. Now, how to understand those networks? So I think you have gone through in the Interactomics course, students who they have gone through all these techniques to a greater extent, which we have the protein networks and the PPI one in the experimental method is to have to hybrid that tandem affinity purification or the co-IP kind of experiments. Whereas in the computational method, you have the domain interaction, orthographic interaction to understand or predict that whether this protein and protein is doing or it is having any kind of association or not. So now we will be looking at the two tools which is available online. One is the web sort and another is free with certain kind of genes to find out what are the interaction levels are those. Yeah, so this is the interface of web default. So what we will do is that you can go here and here you can change your organism of interest, which we will do with the Homo sapiens. So these are the genes which you can get through your mass spec based experiments and through your different high throughput techniques or whatever you use, that which are coming to be as a differential regulation or differential expression in your disease condition, and you are now trying to find out that how they are clustering over a network and what is their association between those things. So now there are three different things, but you can go through over representation analysis or ORA. So we can so you can see that the gene ontology pathway network is what we can uh, get through it. So we will try with the network. So we let us. So we will go with the pathway. And we will keep all this as uh, so we will just reference it is the genome protein coding, and we will keep all these things as default and top 10 significance level. So it takes a few minutes. So I have kept the result ready beforehand as only. So see, you can see this is the enrichment result. So we have used for the network is the PPI biogrid, that is for the protein-protein interactions biogrid. So this is the size for different biogrids. These are IDs which they mention, and they are expected, and the ratio and the p-values and the MTR and the gene symbols what you can get. Also, meanwhile, might be that you can see that yes, that result has also come across. So this was the random genes which I had selected from one of those data sets. So you can see that thyroid cancer is signaling pathways and all those things are coming as enriched with this kind of genes or the protein. But now how these genes interfere or the genes look like or interact with them. So this is another tool that is string. So just type string, and this is functional protein association network. So you just click on multiple proteins and give the list of the name of those proteins. Just a minute. So these are the Uniprot IDs, and organism is Homo sapiens. So if you keep it as auto detect as well, or you can mention it as, because if we are dealing with the Homo sapiens, and then so this will map with the protein from the Uniprot ID with the genes, and then if you just press continue, So you can see this is the network between the different proteins and the association which is coming. Now there are different settings which you can change, like the full string network or the physical and the subnetwork, or on the basis of the evidence, or on the basis of the confidence. The confidence, the more evident it is on the number of publications it has been reported. There is an interaction between the two proteins, like the experimental evidence, like the your experiments, like the yeast two hybrid systems or microarray and other stuff. So it will be more. Also, the what is the algorithm through which it should be extracted out. What is the confidence level required? Highest confidence, high confidence, or the medium confidence that you can adjust. So we can keep high confidence or highest confidence if we want it in a more stringent level, and the different interactions, and you can update and it will come across. Which also you can export as different node to node annotations as a image, as well as you can use it in your different other software like Cytoscape and all to interact, to make it more visually appeal and for using it in your publication purpose. Yeah, so that was all from my side regarding the interaction network. A little overview of how we can do that.
All right, thank you, Ankit. And I think this is an area where a lot of tools are available, and one could actually do a lot of analysis to make the most biological sense of the data. I hope if there is any query coming to the portal, we can take that. But otherwise, let me just kind of give you an overview of not only like just today's session, but in general, why we are talking to you about too many new technologies, right? So whatever areas of life science you are working on, these kind of omic technologies have a lot of potential to bring the new leads, new hypotheses forward, right? Because even if you're not very sure about starting a problem, and what type of targets you're looking at, these omic technologies have potential to bring out the best potential targets, which have to be further, utilized for validation and biological characterization. And more you are able to integrate the data from different type of omics approaches, your hypothesis could be much more refined and you can have much better understanding of the biological pathways. So that's what is shown here, like, you need to be unbiased, but I think in this courses, what we have been more focusing on about is the proteomics and integrating the proteomics with other type of omic technologies as well. More specifically, if you think about clinical situation and the medicine, that's where these technologies have already started making a lot of impact. And the field is going to progress more and more in the future is about personalized medicine. Can we look at one individual as a unit and try to provide as much information as possible from these type of omic technologies, and especially proteomics and metabolomics, to look at the functional molecule, which can help for a given individual to look at the multi-marker risk profiling, looking at disease diagnosis early, looking at the new therapeutic targets. Even if somebody is already having a disease, but they are being given some treatment, some drugs, can we monitor the response of that given drug over the time period? And again, just by looking at the serum sample, plasma sample, looking at a urine sample, one could do a lot of these monitoring, as a part of the routine screening. Also, like, if you can integrate that one individual's information with this clinical data, one could then try to also make some sense of, from the healthy individual as a wellness, or from diseased individual to look for the better treatment of that particular patient. But as I said, like, these technologies are not very specifically used only for the medicine or the clinical field, but also are equally open for the other spheres of life science technology as well. So whatever field all of you, the students of more courses, are going to work on, I'm sure, over the period you are going to find them quite helpful to you, because we are going to be now much more broadening your horizon to think about how to address a given problem much better way. In which way you can now utilize the right tools to look at whether you are looking at understanding the function of it, you are looking at the characterization, you are looking at some sort of the detection, or even the looking at therapeutic or the pharma responses. So many of these tools which we talked about, whether the SPR, BLI, mass spectrometers, microarrays, you will find them being utilized at some or other stage of the workflow. And again, the courses were meant to broaden the scope of it, giving you the opportunities to appreciate the pros and cons of these technologies, so that you can be better prepared in the future to take this knowledge forward and ask the right questions in your given project. I hope you're enjoying the MOOC courses, and soon you will be finishing your exams and you will be given this course. But from our side, if you will be the topper of these courses, we will have some interaction further with you and see in which way we can get you on board at IIT Bombay with some opportunities for the hands-on or even the future workshop or the conference. So please stay in touch with us, and all the best to everybody for your exams. Thank you very much all the TAs for preparing the live sessions and throughout in the course. Thanks everybody and have a good evening. Bye-bye.